Hi there, I'm Susan Lindquist. I'm at the Whitehead Institute in the Department of Biology at MIT, and I'm here to tell you about protein folding. Protein folding is a universal problem of biological systems, and it winds up influencing every aspect of biology that you can imagine. So these are very simple organisms called the yeast Saccharomyces cerevisiae. It's a microorganism. This is obviously a very, very large magnification of these organisms. That organism is responsible for beer, bread, wine, all kinds of things that make life worth living. Anyway, that organism is also a wonderful experimental system that has the same types of problems with protein folding that those organisms over there have. This is a universal aspect of life. And we're used to thinking about life in terms of all the very different things that make up different individuals. But there's a unifying principle of life that relates to protein folding and that plays out from this organism to that organism in ways that allow us to deeply understand some of the worst problems in human biology and try to find clever solutions to fix them. So proteins are us. Many people think about proteins as being food, but the reason why we think of them as being food is because we need to take some of those elements of proteins into ourselves, chop them up into little pieces, reassemble them into our own proteins, because proteins do just about everything that you can think of in our bodies. Proteins are the muscle that powers our arms and legs. Proteins are carrying pigments in our eyes, and it's when light strikes those pigments, it causes the protein to change shape. That sends a signal to our brain, and that's how we see. Proteins are parked in our stomachs and ready to receive the food we eat and tear it apart into little component parts that we can use to build up new proteins that, as I mentioned, do just about everything in our biology that we think about as a living system. Now, the problem with protein folding is that these things look kind of complicated, right? And they are very complicated, but they start out very simply. So the code of life is often called the double helix, and it's a very long linear string of information. It itself doesn't look too interesting, but along different parts of this, it codes for the essential elements of proteins that make up living systems. And a good analogy for the way in which this information is encoded in this long linear molecule is to think about cassette tapes. Now, cassette tapes were something I played all the time when I was young. I know that you're mostly playing CDs and other digital forms of music. But the cassette tape provides a really great analogy for the way in which very complex information can be encoded by a simple long thread. So here we go. Here's a cassette tape. You look at the tape that's wound around those two spools, and it doesn't look the least bit interesting. But when you put it into this machine that decodes the information, just like DNA is decoded in the machinery of the cell, out comes the most amazing and complex and beautiful sounds. You can play another piece of the tape and get completely different sounds. And another piece of the tape. So how does that complexity get encoded in that simple linear molecule? Well, that's a problem biologists have worked on for a long time, and we understand how the code gets decoded into particular elements. There's one particular part of it we still don't perfectly understand, and that we need to understand because it impinges on all aspects of human biology and medicine. These proteins need to fold up into very precise shapes in order to do anything interesting in the cell. And those shapes are incredibly complicated. So let's look at some real protein structures. You can see each of these different parts of the code has been decoded into long linear string, but that folds up and moves back and forth on top of itself. And it's the complexity of this fold that can actually do something powerful. Now, the difficulty in terms of how this plays out into living systems is we don't really understand the forces that allow the protein to fold so precisely into exactly the right shape. What we are understanding, however, is that when they don't fold into exactly that right shape, disaster occurs. And a way of thinking about this, again, to kind of to illustrate this process, is to think again about that music, that long linear piece of information in a cassette tape that encodes extraordinary music. Well, it's played by instruments, right? And the instruments are together, playing together, and they make great music. Just like the proteins are together in a cell, and they make wonderful, wonderful biology. And you have different proteins making different types of biology in your digestive system, in your brain, in your heart. The problem is that making these complex folds is very much like taking a long sheet or square sheet of metal and moving it into forming a musical instrument. So if you get the fold exactly right, it can play some beautiful music. But if there's some very small element of the fold that's you get almost right, but you don't get it quite right, it can be a disaster. Here's another example. A different piece of metal folding up into a different shape, making and doing different things in the cell. But if you don't get it just right, the fold isn't quite right, it's a disaster. So now you've got this orchestra of proteins that make up the living system, and they need to play together exactly right. They need to fold properly, and then they need to play together exactly right if you want the living system to be disease-free. So let's take a look at how these protein instruments are actually functioning together inside of a living cell. You're going to see pictures of proteins serving as architecture, the structural integrity of the cell. You see proteins talking to each other between cells. You'll see proteins cutting proteins, assembling into various structures, disassembling, and all of these are part of the orchestration of life inside the living cell. That's a very special kind of protein that's moving along the information highway of the cell and bringing packets of goodies from one end of the cell to the other end of the cell. Anyway, you can see, I think, the extraordinary complexity of living system and the proteins that are operating in it to keep us biologically active and do all the amazing things that we can do. I urge you, by the way, to get on the web and plug in that reference and take a longer look at the movie. And there's also an animation that will tell you exactly what you're looking at, all in various parts of it. I've just shown you a very small snippet. But this incredibly complicated biology that's represented by this beautiful movie is misrepresented in just one particular way. And that is that in order to illustrate how these proteins are moving about and doing their things and interacting with other proteins in the cell, they've taken most of the proteins out of that system so that you can see them. In reality, the cell is much, much, much more crowded. So you think about this complicated protein fold and the fact that different proteins have different folds. They have to go from a long linear string of amino acids into those complicated folds so that they can interact with themselves properly. And they have to do that in a really crazy environment. They have to do this in an environment that's this packed with proteins. So each one of these colors actually represents a different protein in its complex, beautiful shape that can change and move around and do various things. But this image, although it represents the crowding of a cell, is missing one other piece. By the way, this is a beautiful movie by Adrian Elcock. And again, you can find it on the web. The thing that this particular image does not convey is how energetic the system is. Proteins are actually moving about like crazy all the time, and it's this aspect of proteins being able to move and signal and from one end of the cell to the other end of the cell help us to interpret what we see one moment, and I'm looking here at you or looking over here at the screen. I completely change everything I understand about what I'm looking at because the proteins are changing shape so fast. So living systems have proteins that work at incredible speed. And here's an example of the way they move about and how the crowding is jostling and banging into each other all the time. The one way in which this animation doesn't quite convey what's happening in the cell is that it too, for the purposes of clarity, has been modified in a certain way, and that is it's been slowed down. So just as the other movie I showed you, this was not very crowded and things were moving around rather slowly. In this movie, which shows the crowding, things are actually not moving at real speed. So you can illustrate and understand and look at how these proteins are interacting with each other and moving around. In order to get a realistic idea of how fast these proteins are moving around in the cell, you'd have to speed that movie up not 10 times, not 100 times, not 1,000 times, not 100,000 times, but 1 million times. So that movie is real slow motion compared to what's happening in the biology of a living system. And there you have the heart of the protein folding problem. Because if these long linear strings of amino acids have to fold up into these very, very precise shapes without getting into trouble with other proteins while they're doing it, under such incredibly kinetic, energetic conditions, you can imagine that sometimes they get that fold wrong, just like those musical instruments. If you wouldn't fold the metal exactly right, it could ruin an orchestra. The same thing can happen in living systems. So I want to give you one more illustration, one more analogy. What happens when proteins start to misfold and bang into each other in inappropriate ways and stick to each other, and it's a process we call protein aggregation. And you know exactly what protein aggregation is like. I know that you've seen it many times. The egg white in this little photograph is actually a solution of protein. And the proteins are all folded properly, and so they're clear and beautiful, and they're not in any trouble. But when you apply heat to that system, the proteins start to move around a little faster. They start banging into each other. They start unfolding a little bit. And what happens is the properties of the biological system change completely. And that is, in fact, what you get. So those are aggregated proteins. And it's just a nice visual illustration of the problem that can occur when proteins don't fold properly in our living systems. So we want to understand how we can keep the proteins looking more like this, and how if they start to go off pathway and start to form little bitty aggregates, we can bring them back to life. Because just a little bit of that aggregation state causes disaster for a living system. So what are the solutions that life has found? Well, the first way we started to discover and learn something about the solutions to that problem actually involved heat. So here we have very simple experiment that was done with yeast cells. We're growing yeast cells in a culture in a shaking Erlenmeyer flask, and we took some of those cells out. We took 2 identical aliquots of cells out. What I mean by an aliquot is just a little portion of the culture. So we took 2 identical portions of the culture out. And that one on the top there was exposed directly to a high temperature. And the proteins denatured and killed the cell. This one on the bottom was first exposed to an intermediate temperature to allow it to kind of condition. Basically, it was exposed for half an hour to 39 degrees instead of being shifted directly to 50 degrees. And as you can see, that short half-hour pre-treatment provided tremendous increased capacity of the cells to survive that second higher heat treatment. Now, this is a universal property of life. And so it is not only true for yeast cells, it's true for Arabidopsis seedlings. This is basically the same experiment. Arabidopsis seedlings were planted in these little dishes, one treated directly at high temperatures, the other one given this intermediate treatment that allowed it to condition itself and then to withstand the rigors of that more intense condition. And these are human cells in culture, basically the same experiment. You can do this experiment with all living organisms on Earth because all living organisms face the same problem of protein folding, and they all prepare for problems in protein folding the same way, by making other proteins that help the proteins to stay in their normal shapes and sizes. So how do we find out what they're doing during those conditioning pretreatments? What we do is we, again, take out a small portion of the cells and label them with radioactive amino acids so that as the cell is making its own proteins, each of those proteins will get radioactive amino acids incorporated into it. That allows us then to visualize it, what's happened. We spread those proteins out on a gel, and we put a film on top of it, and wherever there's radioactivity from a newly made protein, we can see the imprint in a line on the gel. And so you can see that at 25 degrees, the normal temperature for this organism, it's making one group of proteins. At 39 degrees, it started making a whole bunch of other proteins. And the sole function of all of those proteins is to cope with this protein folding problem, to help other proteins in the cell maintain their normal shapes or to get rid of them when they've lost their normal shapes. Now, this is a very broadly used survival response. We first started working with it with heat because that's an awful simple manipulation to make within the laboratory. But it turns out these same proteins provide protection against all sorts of difficult conditions, changes in pH, changes in the energy balance of the cell, changes in osmotic strength, many different changes in the cell. In fact, these proteins are constantly being made in smaller amounts over and over again to help cope with this very broad problem in protein folding. So this very broadly used survival response, as I showed you yeast cells, I showed you Arabidopsis seedlings. Arabidopsis is a small little mustard plant. I showed you human cells. Every organism on Earth is making very highly conserved, very similar patterns of proteins under these stress conditions. And it turns out that that plays into human biology and medicine in just an extraordinary variety of different ways. One of the major reasons why we want so deeply to understand this problem is that it drives many aspects of human disease. So one of the things that it drives is the process of infection. When organisms come into our body, and you can see here we've got a fungus that is growing under normal conditions, not inside the body. But when it starts to grow inside the body, it senses this change in temperature, and it starts to realize that it can invade the biological system. And the only way it can do that is by making new proteins and by making this survival response that allows those new proteins to fold properly. So that's one aspect of disease biology that uses that survival response all of the time. Here's another aspect of human biology that uses the survival response. So what you have in blue is normal proteins, and what you have labeled in brown here is the master regulator of this survival response. And that's normal tissue over there. And you can see that the survival response protein is kind of tucked away in little corners of the cells because it's not being used on this normal biological system. But in the cancer cells, you can see that that master regulator has been amplified a great deal. It's actually present now in the center of the cell, and it's directing a whole new program of gene expression. Whole new sets of proteins are being made at the dictum of the cancer cells to help protect those cancer cells and drive the malignant state. And neurodegenerative disease, these are two brain sections from a normal person and from someone who has died from neurodegenerative disease. And what you're seeing here is the devastation wrought by misfolded proteins in the brain. So it turns out that in terms of human beings, we are a bit between a rock and a hard place with regard to this problem in protein folding, because cancer cells and infectious organisms are using their survival response, this heat shock response, to kill us because it strengthens them, it allows them to survive the rigors of a living system. And our brains, conversely, are not using the survival response when we would normally think they should be, because when we die of neurodegenerative diseases, it's because proteins have misfolded, misfunctioned, and just like those instruments that are not playing right with the orchestra, they're causing devastating disease. Now, this puts us in a difficult position, but it's not a place where we can't move and we can't do something important in biological experimentation. And the reason why we can do things that will help to fix these problems is because it is such a universal problem. And so we can take these very simple organisms here, these yeast cells, and understand more about how the biology of that system is driven, how to correct protein folding and how it goes wrong, in order to help these people over here with all of those different protein folding problems I just mentioned to you. So I'll be talking to you about that in the next lectures.